So thank you very much for the invitation. It's a great pleasure to be here. And I would like to continue on this topic of cross-module integration by telling you a few words about the study of consciousness. I think just a few years ago, it would have been difficult to talk about this topic, and the word was just essentially taboo in cognitive science and neuroscience. And there was this idea that neuroscience is limited to the study of objective events, such as sensory motor transformations, an idea that goes back to Descartes, and that the subjective aspects would be escaping scientific investigation. There were philosophers making this argument. But of course, what Christophe started with Francis Crick, and that what many other people have followed up, is the notion that in fact there is no specific philosophical difficulty, there is an experimental difficulty, but we can try to use the tools of investigation of the brain, including brain imaging, to look for objective events that correlate with subjective perspective from a particular person. So what we can call signatures or correlate of conscious processing. And this is exactly the research path that we've been taking in the lab. I think when you have this research program in mind, you have to consider that introspection plays a specific role. And introspection has a bad reputation in cognitive science, but in fact it is indispensable here because it is defining the very phenomenon that we would like to study. It provides the primary data about consciousness, and we need to quantify it and confront it to physiological recordings. And in the end of the talk, after I describe to you this approach from subjective to objective, I would like to ask whether this knowledge can be applied to patients with impaired consciousness. So a very quick definition. First, the problem of consciousness is not the problem of the whole brain. The problem of consciousness differs from the problem of perception because we know that there is a lot of perceptual processing that goes on without consciousness. So the problem of consciousness is really to ask what additional events occur when the subject claims to see a stimulus as opposed to the non-conscious aspect of seeing. And in order to address that, we need, of course, paradigms that allow us to make a visual stimulus, for instance, come in and out of consciousness. And the important advances of the past, let's say, 15 years of research of consciousness has been to realize that we have a lot such paradigms, that we are, in fact, quite able to manipulate consciousness of the subjects at will and to measure it. I will talk only about one such paradigm, which is the masking paradigm. In this situation, we can flash a word or a digit, like the word radio in this example, and the fact of flashing it is not enough to remove it from consciousness, but if we precede it, and especially if we follow it with a mask, we can make it completely disappear from your consciousness. I will try to make a demonstration of this because I think it's fun to see the actual stimulus that we've been using. So I'm switching to this experimental program, which is the exact same that we've been using in the lab. So what you will see as a digit being flashed to you for 16 milliseconds, followed by a blank, and then followed by masking letters around it. Okay? And if you look at this, and we're going to change the duration of the blank, so at this point you should be able to see the digit, because it's a long lag between the digit and the mask. So I hope you can name it, like 4. Okay? Can you see it? Yeah. Good. Okay, so we make this lag shorter and shorter. So now it's 66 milliseconds, which should be close to threshold, and maybe sometimes you won't see which digit it is. But you should still be able to sort of see it. But now if we move to 33 milliseconds, the digit is just completely gone from your consciousness, right? And I swear to you, there is no trick, it's there. It's present on your retina. There is a digit every time, but you can't see it, unless, like I did now, I made an eye movement and the mask was gone and I could see it. So this is one way in which you can let stimuli come in and out of your consciousness. And if we place ourselves at threshold, then of course we can have trials in which sometimes it's conscious, sometimes it's not. So we have a situation with the same physical stimulus, but different subjective aspects, different introspection. So we combine this with measurements of brain activity, and we are especially interested in the last few years in measurements that show us the temporality of conscious access. So for instance, EEG, magnetoencephalography, we can combine the two, as well as intracranial recordings. So I want to show you a few examples from this research strategy using this exact same paradigm. In fact, we can have both objective and subjective measures. So we can ask, can you tell whether the digit was larger or smaller than 5, for instance, or can you name it? And we can also ask, can you rate how visible it was? Was it seen? Was it not seen? The results of both measures is actually very, very similar. There is a major event as a function of the duration of the blank, which is a threshold-like function. So at a certain point, suddenly people begin seeing the stimulus. And that threshold can vary a little bit from subject to subject, but it is at the same location for objective and subjective reports, suggesting there's a sort of major transition in brain activity. Now we can measure this transition using, for instance, EEG, and the results of these experiments have been that access to consciousness seems to be a rather late event. For the first part of the processing of the stimulus, we can see the entry of the stimulus into the visual cortex. We can see it move forward into the system, and we see what has been described by some of the Bayesian theorists as a linear form of accumulation of evidence. The stimulus is progressing in the system, and the longer the delay that we give it, the more it is able to accumulate evidence in favor of the presence of a digit. But then later on, we see a second event, which is what we've called an ignition, a nonlinear event, which is that you either make it above the threshold or you stay below the threshold. So this is one slide from a study using EEG where we reconstructed the brain activity, and what you're seeing is the distributed pattern of activity at 370 milliseconds. But if we place virtual sources at these different cortical locations, we can follow the time course of activity. And you can see, for instance, in this fusiform gyrus here, which is on the visual pathway, you can see that we can really see these two steps. If you look at this first step, the successive curves are for the different delays of presentation of the mask. And if you see this little green curve and even more the others, we have more and more activation in a linear manner as a function of this duration of presentation. But the green curve is the 33 millisecond trial that you just saw was completely invisible. So we get activation, strong activation actually in the ventral visual pathway in the absence of subjective visibility. Then we have this late event, and you see that this late event is present both in the posterior areas, but as also in the parietal and prefrontal areas, and prefrontal cortex quite specific. This is completely nonlinear. The curves stay at the bottom when it's not seen, and they move higher up in this sigmoidal fashion with this threshold function when the item is seen actually consciously. So a sharp difference between early and late periods, and it's the late period which at the scalp level is associated with a wave that we call the P300, which reflects the subjectivity of consciousness. We've done many of these experiments. I want to show you a movie actually where this was obtained in 2005, a slightly different paradigm, the attentional blink paradigm. What's great about this situation is that we have the same exact stimulus, but on half of the trials the subjects rated it as not seen, they couldn't see it, and on the other half they rated it as seen. But physically it's the same simulation, and in fact if we follow this movie, at the bottom is the difference, so you can see the difference. I'm sorry, the labels are a little bit hard to read. So here we're progressing time slice by time slice, and we've reached already 100 milliseconds here, which is a time where we see a lot of striate and extrastriate activity. You can see this bilateral visual activity here, and you can see that it's exactly identical at this moment for conscious and non-conscious trials. If I continue this movie, you'll see the activation progressing forward in the occipitotemporal pathway, and this is the time of the so-called N170, a major potential on the scalp surface. You can see that it corresponds to bilateral activation of the occipitotemporal pathway. We know digits are processed bilaterally, and you can see that, again, it's the same intensity. You can't tell which one is the not seen and which one is the seen trials. They are not different. But suddenly, at around 200 milliseconds, we'll see this huge divergence in brain activity. And you can see already there is much more activation in the seen trials. Also, it's reaching the inferior frontal regions and inferior parietal cortex. And we see this ignition pattern of brain activity, so that there is really much stronger activation in the seen case. In fact, you can see that the not seen situation, very interestingly, is remaining confined to the temporal lobe. And now it's becoming left lateralized. But you see that there is a much more explosion of activity which reaches into the inferior frontal cortex only in the situation where the subject is conscious. So the outcome of these experiments has been a number of signatures of consciousness, suggesting that conscious access is a late event after 200 milliseconds, maybe 300 milliseconds sometimes. It corresponds to a second phase of evidence accumulation. The first one is linear, and the threshold of access to consciousness corresponds to this later event. you can always quibble about the localizations from event-related potentials. It's not easy to go from the scalp recordings to localization, but we've also done these experiments, like many other people, using fMRI, and this is actually an early experiment where we had the masked words versus the visible words. And we found once again that even completely unseen subliminal stimuli do give rise to activation. These include, in the case of words, this visual word form area, quite unique to reading, that we see in the left hemisphere. So we later characterize this system and show that it is really about the knowledge of the orthography of the word. But you can see that a visible word compared to that will ignite a much larger scale network that involves the frontal and parietal lobes and a set of distributed areas. Using intracranial recordings, we can see that another signature of conscious access has to do with the correlation between these different systems. They become much more correlated with each other, they become coherent with each other, and in particular in the so-called beta band of oscillations around 20 hertz, we can see that they become phase synchronized. Can we, and I just wanted to show these slides to show you that this is not unique to things that are done in our lab. There are many labs now around the world doing this sort of experiment. This is an experiment with auditory stimuli where you can have detected sounds and non-detected sounds when they're presented at threshold. And you can see this huge explosion of parietofrontal activation in the detected case. This is a nice experiment which had to do with conscious versus non-conscious control. This is showing, importantly, that you can have operations of inhibition without consciousness, including activation of the pre-SMRI region, for instance, without the subject being aware that he has seen a stimulus which orders him to inhibit behavior. But again, when you're under conscious control, where you know you've seen something and it is an order to refrain from behaving, then you see again this explosion of additional activity in the parietal and prefrontal lobes. And I just wanted to mention here on the right a very beautiful experiment that was done by Kian Quiroga, and again Christophe started this, where you can now record from the brains of epilepsy patients and even record from single neurons in the brain of epilepsy patients. This is a case of single neuron recordings in the anterior temporal lobe. The neurons are responsive to specific pictures, such as the World Trade Center here, but the firings of this neuron occurs specifically only when the picture was visibly seen by the subject in this masked situation. So at 66 milliseconds, this was close to threshold. I don't know if you can see this, but these spikes only occurred on trials that are blue here because the subject was able to report that the picture was seen and not when the picture was not seen. It's completely all or none and at the same sort of late latency as we could see in the MEG or EEG recordings. So what does it all mean? I think we're in search now of a theory for these sort of signatures, and Jean-Pierre Changeux and I have been trying to work towards a theory of this. The theory we call the global neuronal workspace, after the work of the psychologist Bernard Baars, who call it the global workspace. The idea is very simple, and I won't go into any of the details, but the idea is that there are many specialized systems that process information non-consciously in encapsulated form. They have their own specialized inputs and outputs, and our claim is that what we subjectively experience as consciousness is in fact the global availability of information. It's the breaking of this modularity, because we begin to share the information across the brain modules. And what we call being conscious is when the information circulates in this way. So access to consciousness corresponds to the ignition of distributed workspace neurons, neurons with long-distance axons that allow you to share the information. And we claim, and of course there is a lot of anatomy, especially in non-human primates, to show that in prefrontal and parietal cortices, these long-distance networks are partly proeminent, and they have the right connectivity to dispatch the information across the processes. So this is a sort of cartoon picture where a subliminal stimulus is entering the visual system strongly but is dying out progressively as it's having a subliminal influence on other processes. And conscious processing corresponds to a situation where, on the contrary, there's a snowball effect of a more positive feedback nature, activation climbing up the system, reaching these higher level systems, which themselves amplify their inputs, creating reverberating activation, which can be of a long-lasting nature. So a characteristic of information that crosses the threshold is that it can be maintained over a long time period, and this is very similar to what we call working memory, same sort of areas. So the claim is that during conscious access, multiple high-level areas accumulate sensory evidence, they exchange information bidirectionally. They tend to agree with each other, and when they all agree to each other and they reverberate the information, we cross the threshold and we become aware of a piece of information. Maybe I think I will skip this slide because I'm lacking time. I do want to speak about this interpretation, this application rather, of this sort of line of research now to a really important, indeed, a life or death problem, which is that if we begin to understand signatures of consciousness, we should be able to apply it to the clinic. We should be able to provide better separation between the states of coma, vegetative state, minimally conscious and locked-in situation. And you have to know that it's been estimated that about 40% of patients who enter the clinic and are locked in are not initially detected by the clinician, which is a pretty horrible situation. So we have to provide better definitions, better predictors of the patient's outcome, and hopefully new interventions. I think this is a futuristic outlook, but it's beginning to be possible, and we were all stunned by this paper by Adrian Owen and collaborator from Cambridge, showing that with fMRI, in at least one case out of 60 of vegetative state patients, they were able to detect very high-level organized fMRI patterns of activity that suggested that the patient was in fact aware, was able to understand orders spoken to the patient, and was able to, for instance, imagine playing tennis or imagine navigating his apartment, creating these different patterns of activity that were exactly like the controls. Okay. So this is at least a proof of principle that imaging can detect structured brain activity, which looks like a conscious situation. Of course, in the clinic, I think it's going to be much more useful to have access to these measures using EEG. And we've been working with Lionel Nakache and collaborators to try to develop auditory EEG-based signature of conscious processing based on the previous ideas. We've tried to elicit an auditory event-related potential of the P3 sort, this late P3 waveform, by using novelty. Now, let me play for you this paradigm. Imagine that you hear this. Okay? You have a novelty response at the end of the last sound because it's a novel sound in the context of the previous ones. Now, what we can do is we can play this repeatedly. If I can get my mouse back. Thank you. So what we do is we're going to play it repeatedly over and over again. And at the end of this, we may have a deviant. Okay. Now, you just generated a P300. But that's a very strange kind of P300 because it's a P300 because this global sound was novel, and what was novel was that there was no novelty. Okay, it's a sort of second-order novelty in this situation. Now, we've used this paradigm with the idea that only a conscious brain should be able to have this sort of novelty response because it's a P300, because of our past research, but also because in theory, only a conscious brain should be able to have the span of time needed to integrate the rule which defines these 5 stimuli, and to find that a perfectly monotonic stimulus is in fact a violation of that rule. Well, I'm sparing you a lot of this research, but the finding has been that indeed this late P3-like response to a monotonic stimulus we only obtain in the conscious case. The first part, which is the usual mismatch negativity to just a local deviant, can be obtained in a completely non-conscious patient. In a comatose patient, in a vegetative state patient, we'll still get the mismatch negativity. That's the early non-conscious part. But the second order part, we seem to get only in patients who are conscious of seeing something. And we also have a phenomenon of anticipation. They know that something is going to happen before the end of the sentence, before the end of the sequence, sorry. So we can also look at this period before the end of the whole sequence of 5 sounds, and we can analyze it to look for signs of long-distance communication. People in my lab, Jean-Rémy King, Jacobo Sitt, Lionel Nakache, have used what is called symbolic mutual information, which is a way to categorize the EEG signal and look for mutual information across distant electrodes. And what they have found is that indeed, if you reconstruct the patterns of mutual information across long distances of the cortex, you have a very clear signature which is completely different for vegetative state patients compared to minimally conscious patients and the control subjects who are perfectly neurologically normal. You can see that there is this explosion of long-distance communication, which is clearly characteristic of a conscious patient. So we are sort of pinging the cortex, and both the novelty reaction and the pattern of connectivity of the brain are both characteristic of a conscious subject. We can do automated machine learning to these multiple measures of brain activity provided by EEG, and we can compare them with the clinical labels that are provided to these patients. And the fact that there is a lot of patients at the diagonal means that we are indeed decoding to a large extent what patients are conscious and what patients are not conscious. You may think that the off-diagonal terms are all errors, and they might be errors, which means we're not perfectly decoding. But there is something interesting that I don't think the clinical labels are actually gold standard. It's very difficult clinically to categorize patients. And so when a patient is vegetative clinically, but in fact our decoder tells that he might be minimally conscious or even conscious, what we are finding is that in fact the recovery of the patient tends to be much better. There's a much more likelihood that the patient will come out of this state of being conscious, of being vegetative, and might become minimally conscious. So we think that some of these off-diagonal situations are not necessarily errors of the software. I just want to end by saying that this sort of test means that we begin to have a detector of consciousness even applicable to non-verbal organisms, and I'm thinking not just of rats and mice, but in fact of this non-verbal organism. We'd like to know at what age babies become to be conscious. We have applied exactly this test. It's exactly similar, except we present a face at the same time. This is done by Ghislaine and by Anaïta Basira, and the sound is a little bit different. Okay. These are the new stimuli that are more child appropriate. And we are finding that 2-month-olds already show the second order type of violation that we suggest is characteristic of a conscious subject. So we think that 2-month-olds are conscious. This is perhaps not a surprise, but it means that, of course, we can go backwards and ask whether preterm infants, for instance, at what age they are conscious. We find that it is quite late response, so it's no longer a P300, it's a very late protracted response. It means the brain of the child is very sluggish, and this nonlinear phase may come very late. And we have converging evidence for that from other kinds of paradigms, especially from the same sort of visual masking paradigms that we've used before. So it looks like, as was said this morning, the prefrontal cortex of these babies may be functioning. It's already doing nonlinear accumulation of evidence, but with a very protracted period because presumably the fiber tracks that lead to these higher regions are not yet myelinated. So I'd like to conclude by showing you these lovely pictures of the brain as a flame, basically, that we can measure, and by the intensity of the flame we can give an idea of whether it's conscious or not. Now, the perspectives, I think, are to develop actual clinical devices that can be used at the bedside of the patient. The EEG is perfect for that. But also, I think, to develop theory and simulations in a better way, because the theory, as you see, still is more of a verbal kind of theory. We have done some simulations. We sort of know in simplified simulations that we can get the sort of signatures that I've shown to you, but this is only the beginning, and I think a much more realistic picture would be obtained if we had a more precise simulation of the cortical layers involved. So thank you very much for your attention.